I'm Paul Verschure, and I'm a co-author of a paper with my colleague Francisco Páscoa dos Santos. Welcome, Francisco.
Hello, thank you.
So, Francisco, the paper is called Excitatory and Inhibitory Homeostasis and Diaschisis, tying the local and global scales in the post-stroke cortex. Why is this an exciting title and paper?
Well, there are several reasons, but let's focus first on the diaschisis. Like, this is a term that was already coined in 1913, 1914 by von Monachov, and it describes how local lesions after a stroke can have very widespread effects, which is in itself a pretty amazing phenomenon. But to me, what is most interesting is then how the brain can recover, sometimes spontaneously and globally, from these widespread effects. And we were trying to look for potential mechanisms that would drive this recovery. And after looking at the literature, we found this excitatory-inhibitory homeostasis and its signatures in a lot of stroke research. And so what I thought here is that in the process of trying to balance excitation and inhibition, the different cortical regions that are affected after a stroke could achieve this recovery and correct this diaschisis that is felt after a stroke. And it's still a pretty mysterious phenomenon, and the recovery from it as well. And so we hope to shed some light into it through the lens of excitatory-inhibitory homeostasis.
Yeah. But now, so indeed, it's a very surprising phenomenon, right? It's old, local lesion, global change. And it's very mysterious how this could play out, right? But so the one that is problem of scale, yeah, because we have a local lesion disrupting local networks, and then you have a global change in activity patterns. So how many scales should we consider in trying to understand this?
Yeah, I think in the point of view of this paper, we are mostly focused on the mesoscale, which would be related to local cortical networks and how they maintain their relevant activity patterns, and in this case, the excitatory-inhibitory balance. And then from this mesoscale, we get into a macro scale, which would then be related to the coupling functional interactions between different anatomical parts of the cortex. Of course, you can look at this phenomena at even smaller scales in synaptic level, but these are the two that we most focus on in this review.
Yeah, but so that was a bit my question, right? Is it in the end enough? Should we actually consider more levels, or is this a good approximation?
Yeah, that's a very relevant question, because of course this excitatory homeostasis, although it tries to maintain the population firing rates of neurons in the cortex at a stable level, it's still a form of synaptic plasticity. So it acts on the level of synapses. So we have changes, maybe even in intrinsic excitability properties of cortical neurons. And of course, there are other phenomena that occur post-stroke that have to do more with the neuronal scale. So for example, we have an acute increase in glutamatergic signaling that might be neurotoxic, and concurrently, we have also an increase in tonic GABA signaling, which is thought to be a sort of a neuroprotective mechanism. And these are phenomena that happen on a cellular scale level that might also be interesting. But they are also happening on short time scales, and that's another scaling perspective that we have in this review. We are interested in longitudinal processes that happen in very slow time scales.
Yeah, so this is one of your observations, right? That in some sense you seem to make the argument that we almost can distinguish the fast processes from the slow process, that really their dynamics is almost independent, not fully, but almost. So would you agree with that?
Yes, in a sense, I agree, because if we look at the literature, processes such as the ones that I just explained, and the kind of let's say adaptive homeostatic changes that we see in the long term, they are sometimes really different. So, for example, as I told you, there is an increase in tonic GABAergic signaling, like in the short time post-stroke. That is thought to be a neuroprotective mechanism, but in the long term, it's actually beneficial that you have a decrease in GABAergic signaling to compensate for the loss of excitation. And this is what we think happens through excitatory-inhibitory homeostasis, which indeed occurs in a slow time scale. So there is evidence to at least partially decouple these two phenomena that are important for recovery. And here we focus more on the slower mechanisms.
Yeah. But now the structural primitive that you build on in your analysis is also the local excitatory inhibitory balance, right? So this is where the whole analysis departs from. Yes. So why do you believe that is a proper primitive from which we can start to reason about more global brain dynamics? And then also in terms of detailed cortical circuits, because we speak about the neocortex, what does it map to in a real cortical local volume of cells?
Yeah, okay. So one of the reasons why I thought that looking at this locally would be a good idea is because, well, I come from a background of modeling. And of course, I was trying to study this phenomenon in a whole brain large-scale model. And for that, we often use, to constrain our models, we use human structural connectivity data. And one thing that you can notice there is that there are some regions in the brain, so for example, in the parietal or superior frontal cortex, that really are, let's say, connector hubs, which means that they receive a lot of projections from other parts of the cortex and also send a lot of projections back. And this, from a modeling perspective, what I noticed is that these nodes, these particular areas, receive a lot more excitation than, so for example, parts of the sensory motor cortex. So, and since they receive significantly more excitation, it is logical that after a lesion, and also depending on the localization of the lesion, they will require a different level of compensation to recover their balance. And what's interesting as well is that by also accounting for this in the local level, we can see the heterogeneities in this homeostasis after certain lesions. So we'll see that certain parts of the brain will require greater changes to achieve recovery in terms of excitatory inhibitory balance.
But before we go there to recovery, I would like to drill in a little bit more on this local circuit, because on the one hand, not all excitation and inhibition is created equal, right? We have different inhibitory neurons that work at different time constants in different layers of the cortex, and the same for the excitatory neurons, right? So how should I map the excitatory inhibitory balance to, let's say, a cortical column? Which neurons would we really be talking about in a cortical column in your view?
I see your question more clearly now, and I would say, in general, from what I know from the literature, these types of changes happen in most pyramidal neurons of the cortex, from layer 4 to the superficial layers. However, when we are talking about inhibitory neurons, this has mostly been found in parvalbumin positive interneurons. So the role of other types of interneurons, so somatostatin, VIP, is still somewhat unclear. this phenomena, but what research shows us is that mostly this homeostasis is performed, or it takes place at the level of the synapses from parvalbumin positive to pyramidal internerve.
Right. Cool. Okay, but then also as a knock-on effect of the lesion, also the thalamocortical interaction will change. In some sense, now we have a compromised cortical circuit, which is lesioned, and there are inflammatory factors coming in and what have you. And in the meantime, the thalamus is switching also into a pathological state. So it starts to pump, let's say, low frequency bursts into that disrupted cortical sheet, right? Pumping a lot of energy into it. So do you think that also is a contributing factor in sort of driving then this kind of change of the circuit? Or in your analysis right now, the thalamus, you sort of leave out of that discussion, or at least you're thinking about it?
Yeah, so in this paper, we focus mostly on the cortex, but that's definitely also an important part of the whole process. You also know that in one of the papers from SPECS, it has been shown that thalamocortical networks enter this state of dysrhythmia precisely because the thalamus loses a lot of excitation that's usually coming from the cortex. And that's because the cortical neurons are not only lost, but the ones that remain are underactive. And through this type of excitatory-inhibitory homeostasis, one of the goals is to recover the activity levels of the cortex back to normal. And we expect that through that process, the thalamocortical dysrhythmia might also be a bit inhibited, well, back towards the non-pathological pattern of activity. Yeah.
But now we have to work our way towards the diastasis we talked about earlier, right? And that also means we have to think about the intra and interhemispheric impact of that local lesion, right? So how, if I have this local disruption, how do we explain the spreading of it globally and also to the other hemisphere?
Yeah, that is a very difficult question. You know, people have been working for many years trying to figure out how these effects spread, but I think we have to look. One of the things we have to look at is how the human structural connectivity, well, is structured. Sorry for the pleonasm here, but it has certain graph properties that make it not only robust to this kind of lesions, but also in a way fragile. And because we are looking at the functional interactions, what I believe might be very important to consider in this case is that for you to have a functional coupling between two parts of the brain that are in different hemispheres, they must not show abnormal patterns of the activity. So they must be able to oscillate and co-oscillate in a healthy manner. And what happens is that in one hemisphere, if you lose that ability, even if in the other hemisphere you might have an also pathological activity, it's hard to couple these two regions. That might be one of the reasons why we see these effects of stroke spreading across the whole brain. And of course, again, by recovering the balance and bringing back these normal patterns of activity, you would be helping the recovery of this coupling between the two.
That's what I wanted to go to next, right? Because now we know the local circuit. We have an idea why this is spreading also to the other hemisphere, even though there's a lot of stuff we can still analyze there. But now you're saying, actually, there is a trick. The brain is deploying a trick to, if you want, renormalize these interactions. And this is done through homeostatic plasticity. Right, so what do you exactly mean with that? Maybe you can explain a little bit what's homeostatic plasticity and why would that lead to a renormalization?
Sure. So there are several types of homeostatic plasticity in the cortex in the context of excitatory-inhibitory balance. But well, one of the most studied is synaptic scaling. And this is a process through which, for example, a pyramidal neuron that has just lost a lot of its normal input or has experienced some perturbation in its normal input. And to bring back activity to the pre-perturbation levels without messing up the relative strengths of the synapses, which are storing important information, the neurons will scale equally all of their synapses so that they can maintain the normal patterns of activity while not perturbing the relative synaptic dynamics that help store information. And this happens at the level of excitatory to excitatory synapses, inhibitory to excitatory synapses, which might be actually a very relevant process in stroke recovery. But in short, it's a way of neurons to maintain a healthy level of activity. Let's put it like that.
Okay. Now you're provoking me because you used the word healthy, and that's normative. And in some sense, the idea of a homeostatic process also is a normative concept. That means there is some set point, right, that you strive towards and you want to keep stable at that set point. Mm-hmm. So what is that set point? And why would that set point be so important for, if you want, the information architecture of, in this case, the neocortex?
Yeah, that is a very relevant question. And it's a question that people have been trying to answer. But there are some hints that came in a very recent research paper where people took data from the visual cortex of mice after sensory deprivation. And they were trying to figure out, okay, the network is trying to recover an healthy or normal firing rate, but what does that mean? So they looked at this, let's say, activity regime that is thought to be also quite important to the brain, which is criticality. And criticality is related to a state where a network of interacting units is poised near a point of transition between, let's say, stable low activity dynamics and, for example, oscillations. And this is what is called a bifurcation. And if the network is poised in the point right before the bifurcation, this has been shown to optimize a number of functions from optimizing the dynamic ranges in response to stimuli, information transmission and storage. And I mean, there has been a lot of research making the case that criticality is an important functional property of the human brain. And what they've shown in this study that I was talking about is that through this excitatory-inhibitory homeostasis, not only are the firing rates recovered, but also the networks are brought back to this regime of criticality. And that is a hint of what this set point might be and why it is functionally relevant. Mm-hmm. And that is also something that we mentioned in this review.
Right. But so this is cool, right? But now we have something about, let's say, the information architecture and criticality for the healthy brain, why it will always be tuning, poising itself at this edge, also the edge of chaos, if you want. But now, how does it help us to understand pathology, right? Because diaschisis is a pathological pattern. It correlates with functional deficits. It correlates with certain also manifestations physiologically. So how does it now help us to understand those pathological aspects of brain dynamics?
So, for example, one of the ways where I think this criticality could be relevant is that when you have a critical system, you observe certain types of transient dynamics, where the system would jump from its state of low activity to oscillations and remain there for a certain time, and then come back to its low activity. etc., etc. And these transient states, they do not have a characteristic scale. It's a property of a critical systems that if you plot the distribution of the sizes of these events, it's a power law. So that it's a line in a log plot. And this type of transient activity is also something we see in our fMRI bolt signals, which is what we then use to infer functional connectivity or functional coupling between regions in the brain. So the idea here is that if individual regions are not able to maintain this type of dynamics, they would have a more difficult time, coupling with each other transiently in patterns that resemble the functional networks that we see in the human brain. Mm-hmm. And so after a stroke, an effect of this criticality, which is affected locally, would also be a disruption in the ability of brain regions to communicate with other parts of the brain. And this is very strongly observed in these interhemispheric functional connections that you mentioned before.
Mm-hmm. So then, in some sense, you would say that the brain tries to poise itself at this point of criticality, and then a brain that's affected by a lesion, as in the case of stroke, would be subcritical or supercritical.
I feel that there is a lack of studies to give us information on that, but the intuition is that it would be subcritical, because by losing excitation from the immediate effect of lesion and losing all the projections that come from the part that was lesion, or even cutting white matter tracts that are providing this excitation from one part to the other, I mean, you are basically losing a lot of drive, so it's easier to be stuck in this subcritical state. That is my intuition.
But also, what we should consider is that it might not be homogeneous in this distribution. Right? Maybe the healthy brain has a more homogeneous or gradient-like control over criticality. For instance, reflecting this known gradient from posterior to frontal, to anterior. And maybe that's disrupted. So you have a very fragmented distribution of different criticality characteristics across the cortex. This might be an alternative, right? Now,
no, absolutely.
A key figure in your paper is figure 4, which is in your background. And this sort of summarizes your view on how excitatory-inhibitory balance and homeostatic plasticity could also lead to, if you want, functional recovery in some sense. So maybe you want to step us through those 3 stages that you distinguish there, and how this actually, how we can try to also validate that model that you propose here.
For sure. So I realize now that it's missing the label here at the top, but I will walk you through it. Basically, this is a schematic of we start with the healthy brain here, and this is what we would understood as a healthy functional connectivity. So we have the different parts of the cortex represented by the dots, and the edges represent the functional connections. And so we would have this healthy network with certain properties, modularity, small worldedness, that are characteristic of a healthy brain. And then inside each of these nodes, then we have our local cortical circuits, which in a very simplified manner are composed of interacting excitatory and inhibitory populations. And this would be our healthy situation. And now okay, so this would represent the acute phase of stroke. So you have a lesion, which means that you lose this part of the brain and the respective connections it has to the rest of the cortex. And what happens here is that these other parts that were connected to the lesion will lose a lot of excitation. So while their internal synaptic organization still hasn't had time to change to respond to this, they are receiving less excitation. So that's why we consider there to be an excitatory-inhibitory imbalance. And then simultaneously, we see these other changes that we that are common in acute post-stroke patients. So decreased interhemispheric interactions, and in some cases, increased intra-hemispheric interactions. And what we think is now happens now is that during the process of recovery, which happens in the slow time scale of weeks, months, the local circuitry of the cortex will have to adapt to this excitatory-inhibitory imbalance. And this happens, for example, by increasing the strength of recurrent excitatory synapses or decreasing inhibition. And this has been particularly the case of decreasing the levels of inhibition in a longitudinal way. This has been observed in actual stroke patients and shown to be correlated with recovery. And simultaneously to these processes, we also see a recovery of functional connectivity properties towards healthy levels, sometimes through the formation of new cortical connections, which happen at different levels, but it's what we call cortical reorganization. And our point is that these two processes not only happen simultaneously, but in a synergetic manner, so that the excitatory-inhibitory homeostasis is a key driver of this functional recovery. That was mostly what we're trying to say with you.
Right. That's fantastic. So thank you. And then, but would you believe that this view on recovery would also generalize to other neural systems outside of the neocortex, let's say in the basal ganglia, the cerebellum, hippocampus? Do you think the same principles will hold there as well?
Not likely, except for the hippocampus, because as you know, the hippocampus has a structure that is in a way similar to the cortex. It's as if it is a mini cortex.
It's a proto-cortex.
That's
where the cortex started evolutionarily.
And this same type of homeostatic mechanisms are known to take part in the hippocampus as well. So that is a region where this might take a relevant part as well in recovery. Then for regions like the basal ganglia, striatum, where you have mostly inhibitory neurons, I find it more difficult to include this type of mechanisms. So I would refrain from making very strong comments about that.
All right. But okay, it's something to pursue. Now, two questions to finish up. So what do you see as the most critical experiment that must be performed to test this hypothesis?
So one of the ways that we could do this in two ways. One of them is modeling, because when we use this type of large-scale modelings, we are working in, let's say, a very constrained environment. We can apply the lesions we want. We can measure things at different levels as well. We can measure things at the local level. We can extract fMRI surrogates to calculate this functional connectivity. So one possibility would be to apply lesions in large-scale models that have these types of mechanisms, and then look at the long-term consequences and how they adapt. Another option is to, this requires more time and of course a bigger effort, is to have longitudinal recordings of stroke patients and measure this functional connectivity, and at the same time approximate their excitability through other types of electrophysiological data. For example, EEG. People have been working on ways to measure criticality from EEG signals as well. But what is important here is to do this longitudinally and simultaneously, so we can correlate these two processes. Mm-hmm. Those are two ways it can be done.
But also by emphasizing this computational perspective, right, which seems also a necessary methodology to cross the levels, right, and bring the levels together and really extract and validate the computational principles. This, in some sense, also is a step towards personalized computational neurology, if you want, right? Because now we can start to build individualized models for each patient, include the lesion, and predict based on your ideas about homeostatic plasticity, how the diaschisis for those patients, or also the reshaping of their cortical graph, will take shape and be driven. Do you also see that as one of the next steps that you can pursue with in this line of thinking?
Yes, absolutely. And the beauty of models to me is that you just take some type of generative mechanism and you put it in a model. You observe some patterns of activity or structure that you consider to sufficiently represent what we know about the human brain. And then, yes, we can, for example, apply specific information from a stroke patient and try to predict what's going to happen in the future. And I think even more interesting than that is that we could potentially test therapies for stroke in these personalized models. So, for example, if we see that for this particular patient with this particular lesion, there will be a higher tendency of the temporal cortex to lose excitability, we know that we, for example, might have to potentiate this excitability or this excitatory-inhibitory homeostasis in that particular region for that patient. And also be able to test for adverse side effects directly in these models as well. So
yeah, that's a fantastic potential that you now sort of also point towards in the domain of stroke and stroke rehabilitation. So look, I think I'm really looking forward to your next manuscript, Francesco. This is a fantastic door you have opened here, and I think a very solid review. Now, of course, we just got to do it by building those models and finding also then the clinical validation of them. So thank you very much for this conversation. And
thank you.
Looking forward to your next paper.
Yeah, thank you, Paul. And if you don't mind, I would also like to ask a question to you.
Oh, okay.
So yeah, given your experience in this world of neurorehabilitation, neuroengineering, so going beyond the modeling of this phenomena, where do you think the future of this rehabilitative technology resides, together with these models, in order to, bridge these two approaches to give more precise rehabilitation to patients? So, what kind of technological improvements do you see in the future for this field?
In some sense, it was also a bit embedded in my previous question. In the rehabilitation or the diagnosis and especially prognostics in neurological disease, we have amazing challenges, right? So in some sense, stroke is the simplest deficit of the brain we try to deal with. It's just a lesion. We know where it is. But irrespective of that, we have immense difficulties to predict how such a patient will evolve, what kind of symptoms might emerge on the long run, right? So this is a real void in the clinical toolbox. And there, I believe that the kind of models that you identify here, and also the reasoning behind them, can become a critical tool to make us more proactive in how we deal with neurological disease, right? That we can really anticipate, oh, this patient will probably develop chronic pain in this part of the body because the model tells us that this diaschisis is sort of reshaping the circuit in a pathological fashion. If you know that's going to happen, okay, then you can start to think about functional interventions to prevent it from happening. Plus, we can rethink, as you were sketching, our interventions, because maybe we say, okay, maybe we should, through non-invasive stimulation, drive excitatory-inhibitory balance, so we push this reshaping in a different direction as it would go intrinsically. So I see a massive potential for this approach, really individualized computational neurology that we can start to build now, and that, of course, that we can then link to the kind of functional interventions we are already delivering, because I do believe we can, of course, manipulate brain states, but we can only make that manipulation effective if we also constrain it by functional relevance. You cannot just say, oh, I have an evolving network and I'm going to push plasticity in certain directions, if that's not embedded in this embodied task-oriented function that you actually want to restore in your patient, right? So for me, this is the critical step to get us into the future of really treating neurological disease effectively. So this is a critical step, and that's why, in my mind, your paper is so important. So Francisco, great work. Thank you very much.
Thank you.
And